Okay, morning everybody. I believe we left off Wednesday right at this slide. We didn't talk about it, but this is right at our stopping point. So there's a lot of stuff when it comes to our all of our cells and thinking about surveillance and recognition and knowing this is my stuff or this is not. All right. And this mechanism that we're seeing is part of it, is on the surface of our cells that we're looking at right here. Our nucleated cells, and we've mentioned this previously, the red blood cells don't have the MHC complexes on them, and there's important aspects of that. But on the nucleated cells, we can see specific proteins. On our normal cells, we see the class 1 proteins, MHC class 1 proteins, and remember major histocompatibility is MHC. And when we think about some of the white blood cells, they also have class 2 receptors. So what we're looking at could be something like a macrophage, a dendritic cell, or a B cell. These are the cells that also have this. And this is what lends to their potential for communication, recognition of the foreign substances, and communication about what they have found. All right, it all is basically happening in this complex. Over here, these complexes of proteins are basically our cellular fingerprints that say this is me. All right. And if there are deviations, things like natural killer cells may recognize our own cells missing or misshapen class 1s and basically stimulate their destruction. All right. So the idea with this is truly to give us multiple levels of protection. We're thinking about all sorts, the plethora of foreign invaders that can be targeting us. We can communicate that with this class of proteins. And we can see here the specific subunits, beta and alpha subunits, tethering in the plasma membrane. So they're spanning through the layer. This is protruding to the outside, extracellular fluid, the outside of the cell. And then down below, this would be the guts, the cytoplasm of those cells. So there's all sorts of stuff that we think about in the process of finding something foreign and then basically doing some show and tell, trying to stimulate other things to come in and respond accordingly. So I know the video we showed last week, and that's also posted on Moodle. You can go in there and check some of this stuff out. There are some details describing the dendritic cells and how they're participating in some of these activities. So we're not going to go back and revisit that today, but I recommend, if you haven't looked at that, you take a peek. So the dendritic cells, these guys they're not exactly the same, but they're pretty stinking close to macrophages. Dendritic cells and macrophages, these are both like monocytes that in turn move out of the circulation and are kind of differentiated. So we've mentioned macrophages with the potential for these guys to have a handful of different names based off of where they might be. Dendritic cells basically are macrophages of the skin, all right? We treat them as kind of, separate things really, but it's really just where they're existing, all right? So they've got the potential to involve something, to phagocytize it, to process, to present. They also have some potentials to truly, with this presentation, try to show their stuff to everybody, not really thinking T cells, but everybody that they can, trying to stimulate as much of a response as possible. We're showing you a small aspect. So this bacterium up at the top, we've got some sort of pathogen. All of our little red bits on it, we could think of the antigens or the specific epitopes, the stuff that our cells could recognize as being completely different or super foreign. All right. And that, what we're thinking an immune response is pretty important. The more different it is, the better off we are at mounting some sort of a defense. So it engulfs this bacterium as it happens to phagocytize this. It's within a vesicle, a phagocytome. That phagocytome will fuse with a lysosome, creating a phagosome lysosome. In biology texts, sometimes they call that a secondary food vacuole. Same sort of an idea, basically, as all of this stuff is fusing. So the lysosomes, as we have described before, have the potential for all sorts of digestive enzymes, for substances that are going to truly do a number on whatever it is we've pulled in, degrading it, and then lending to, well, all sorts of different potentials within that cell. There are some other things within the lysosomes that lend to this presentation. Proteases are types of enzymes that recognize protein molecules. So this big mass of amino acids could get chopped up into smaller bits with the proteases, and that's where we get our respective epitopes, the little bits of those antigens that can stimulate an immune response, all right? Just because we've got this chunk doesn't necessarily mean that it's going to be adequate to stimulate a response. There's all sorts of potentials with that. But in a perfect situation, we process all of this, we chop it up into bunches of little bits. Now you've got a boatload of epitopes that can be presented, all right? We're just showing you one. All right, so this, and again, the little red guys on the bacterial cell are what we're going to try to present. So we destroy, there's all sorts of things in here that are being processed and degraded, but it's the red stuff that we're really focusing on, that epitope that we're going to present. So as this happens and those proteases chop this stuff up, we are going to dump some wastes to the outside, but those red bits, the epitopes get processed differently, and we're going to show those on the surface of these cells. So the green stuff in here, these are the MHC2 proteins, and as we showed in the previous figure, you've got a handful of these polypeptides that come together to form this complex. So a quaternary structure, 4 of them coming together to make our active functional protein. All of that stuff would be occurring in Golgi, right? In Golgi apparatus, these respective proteins that were each their own separate bit of mRNA merge within Golgi. We package all 4 of those subunits up together. And in the process of doing that, we also have our little bit of epitopes that have been chopped out, if you will. They fuse with these proteins and in turn get exported to the outside. So it's all happening inside after it's been phagocytized, processed, linking up with the MHC2 protein that's now going to get exported to the surface of this dendritic cell. It would have all sorts of different ones on there, all right? We're just showing you one, but all of the bits that we've been able to process and chop up into things that we think could be potentially big enough to stimulate a response, it's going to be all over it, all right? So studded with different MHC2s, each of which possibly holding a different epitope. Then these guys, the dendritic cells, macrophages would be doing similar stuff. They float around hoping to bump into some T cells. There's cytokines that are being released that stimulate those T cells to be like, hey, something's going on. We ought to see what's up. And the dendritic cells and T cells come together and potentially, if things fit, there will be a communication between the two. We'll see this here in just a second. And basically the dendritic cell is going to exchange this epitope, this antigen, to the T cell, and the T cell will respond accordingly after that. So this is a process of recognizing and then showing or presenting what we have found as being foreign. That stimulates a whole slew of cells to come in and start dealing with things, more than just T cells that want to make some of these initial recognitions. Cytokines being released can stimulate other cells to respond as well, ramping up an inflammatory response, which enhances these cells a greater opportunity as those vessels start to dilate, giving you a greater likelihood that the immune cells are going to be more present and able to get into this area of concern. All right. So this is an electron micrograph, a scanning electron micrograph of a T lymphocyte. So zooming in on it, we showed some stains with some cells, some of our white cells, and you could see lymphocytes, but just from a regular blood smear, you can't tell the difference between a B cell and a T cell. They look pretty much the same. So this image of this T cell, if I imagine if we were to have a B cell up there, there'd be some similarity with that as well. All right. There are definitely some differences, and we can see some of these protrusions on the surface of these cells. These spike like structures are specific proteins that will be lending to this recognition. All right. That's really what is going to possibly interact with those dendritic cells or other cells presenting antigen, and then make some exchange. All right. So before we progress with some of this, we want to think a little bit about these immune cells and really where they're coming from in the first place. All right. So your B cells and others, as we're thinking about the like hematopoietic stem cells generating all sorts of other immune cells, we think of the bone marrow as this site of rich activity. In adults, it's kind of sequestered to just a few bones. As a little kid, there are considerably more areas where we're getting the production of this. And that kind of makes sense as you're young and growing. You want to have more areas that are producing these cells. As you mature, then we're not thinking growth, we're just thinking kind of replacement. And that's a bit different. All right. So it doesn't need as much surface area for the generation of that. So the red marrow is where our B cells are being generated. And we see all sorts of other stuff as well, the red blood cells, platelets, and so on. The T cells, when we think about that, they mature within the thymus. This is where the maturation is happening. And really, once you hit adulthood, the thymus starts to kind of atrophy a little bit. It's much more rich with activity until you're, mid-20s, and then it starts to wane a little bit. You still are, and this is looking at I think it says newborn, thymus of a newborn, as we're looking at this cross section. I saw it, headline, I didn't click on all of the details, but it was a little baby that had a heart and thymus transplant. First, I believe it said first ever. I thought that's pretty impressive. He must have had some serious issues with immunity if his thymus was inactive. Like I said, I didn't read all of the details, but I did think that was pretty interesting. So this bit right here would kind of, as we saw that electron micrograph of the T cell and the protrusions, this is kind of what we're thinking about, but cartoon-like, all right? So that T cell was full of all sorts of things, really receptors, proteins that are there to hopefully do some detecting, right? And we'll move in a minute and think a little bit more about some of this stuff, but there are a handful of different types of T cells that can be doing some of this surveillance. Thinking about some of this initial stuff, we'll want to think about the CD4 or T helper cells. These are the cells that are kind of like the central hub of the immune response. They're the ones that are told this is what's happened, and in turn, they tell the other cells, depending upon what they see, they tell the other cells how to respond, all right? So if there are issues with the production of T cells for one reason or another, like the little dude whose thymus was all messed up, I assume he's not producing any, and his immune response is basically non-existent, all right? How can you do any sort of surveillance when the person that should be telling which cell to do what isn't there, right? That's a huge issue. And we'll hit some more details with that a little bit later, all right? So here is the membrane, plasma membrane, and then protruding to the extracellular area and cytoplasm if we're looking down below. This bit of protein, as we are looking at it, it's not the same, but it is a similar idea to the antibodies that we described before, where you've got a region that's able to do some sort of detecting the binding site that can recognize specific stuff when stimulated, all right? This is a similar, not the same, but similar idea. This class of proteins, this green bit is the variable region that's like the variable region in an immunoglobulin that has the potential deep down to recognize anything, right? But it's not this one cell and they can see anything. It's a plethora of cells, each tasked with their own opportunity to see stuff, right? And that dendritic cell or other antigen presenting cell needs to find the proper cell to see what's going on for these receptors to function properly, right? And truly that to me is fascinating that we've got all of this potential for these cells to make this recognition. So the binding site in green, again, this variable bit and similar as we think about things like immunoglobulins, that really kind of fits with what we think about with the rest of this. So initially, up above our top cell is a naive helper T cell. This cell has yet to decide what's going to happen, all right? And as we move through and see some details with this, one thing I want to point out, and I think I have mentioned this before, we oftentimes talk about things separate. We think about a cell-mediated response, which would be coming over here with this T helper 1, right? We also think about antibody-mediated responses or humoral responses, T helper 2s, right? And we think about them separate, we talk about them separate, but it hopefully makes sense if you are dealing with some sort of infectious substance, whatever it might be, bacteria or fungus or whoever, right? If you're dealing with some sort of organism that's causing disease, you would hope, I think you would hope, that both lines would be stimulated to come in and respond, all right? Sometimes that's not the case, and you do only have one way that you're trying to treat this, but overall, you kind of hope that both of these things are happening, all right? In immunology class, we definitely go through details, great details with what's happening. And we think about them as kind of separate arms of the immune response. And then we bring it all back together and say, really, it's happening together, all right? So keep that in the back of your mind. Up above, the naive helper T cell has yet to truly decide, if you will, what it's going to do. And how it decides is based off of what's happening with this initial presentation, all right? Shall we go this route or that route? Then you get some memory cells. That's kind of after the fact, where we have a few of them, like the B cells producing some memory cells after being stimulated. It's a similar idea where they are there to respond at a future time, all right? So as it gets activated, that's this box right here, all right? And I think maybe we've got another image that shows some more of this, maybe blowing up right at this bit here. So this bit here, this antigen binding site in green that we see on our T cell, that's right in here, all right? And our MHC2 molecule bringing that epitope, I could flip back a couple of slides to that dendritic cell where that's getting processed and presented. That's happening right here, right? So now we've got our little red fragment from that bacterium. That's the epitope that says this is a bad guy, right? The MHC2 protein is showing it, and the undifferentiated T cell makes contact with this, sees it. This CD4, this is another protein that basically helps to stabilize and facilitate the possible exchange. If these are able to interact in that antigen binding site on the T cell, it's going to pick up this epitope, it's going to pick up this protein, and now do one or the other, all right? It's at this junction right here that basically causes this T cell to go from being a naive, unstimulated cell to getting that stimuli and becoming activated, all right? At this point, it now starts telling all of these other cells, secreting cytokines and stimulating things to come in and respond, dependent upon this initial recognition. It differentiates to tell other cell-mediated responses, other white blood cells like neutrophils and eosinophils and the basophils, stimulating macrophages. The T helper 2 stimulates B cells, right? And then again, once all of this stimulation starts happening, we generate some of them that kick back and wait as memory cells for, again, a future infection. So here we could see, this is a cytotoxic T lymphocyte, so little bit different than that CD4. Let me back it up, CD4 or T helper, all right? This, we would say, is a CD8, a cytotoxic T cell, all right? There's different classes of these T cells, right? Not the same, but pretty close to what a natural killer cell is doing, all right? That's kind of the idea, all right? So this naive CD8 cell, and you can see that right here, it's a different docking protein than the T helpers, all right? Helps to facilitate this recognition if what's being shown, and in this case, we're not looking, back it up here, at the class 2s. This is our own stuff, all right? So our own cell with the class 1 protein. Now we've got a cell that's infected, some sort of intracellular pathogen. It could be a virus. It could be an intracellular bacterium. Something's inside of it, all right? And this cell can raise a little beacon or a flag to the outside on that MHC1. And this T cell can see that and recognize this MHC1 isn't what it should be, all right? Something's messed up. And that activates the cytotoxic T cell, cytotoxic T lymphocyte, still a T cell. And like the natural killer cell, it causes the destruction of our own cell, but it's the cell that's messed up, right? It's got something going on inside of it. So numerous enzymes are released, granzymes, perforins. These things basically cause destruction of our own cell membrane and poke holes and chew up. The granzymes are types of enzymes that chew up these substances. You can see little blebs of the cell fragmenting and basically being destroyed through apoptosis, right? So they're not the same, but kind of a similar function as those natural killer cells. And natural killer cells also are detecting issues with the class 1 proteins on our own cell surface, right? Whereas the other route, we're looking at immune cells doing the presentation on the class 2 to stimulate the T helpers, right? So these against, something that's been found as foreign, and the CD8 or cytotoxic T cells are surveilling our own cells, not waiting for a macrophage or dendritic cell to say, look, what's up, all right? It's our own cell saying, come on, help. So here we've got the purple up at the top is a macrophage that's doing the presentation instead of the dendritic cell. So it's presenting an epitope, and down here doesn't match the T cell receptor. And it's, so in this case, the first bit not activated, all right? The little bit in this antigen binding site doesn't match over here, all right? And that's what we're seeing here, all right? So not recognized by that, that's potential, in this case, a potential issue, all right? So next door in this, forget about this first bit, we're not recognizing like this and stimulating. Next, there can be a different opportunity where we don't have that epitope, but we still lend to recognition between the two, but it's not within that specific binding site. We bridge them with the super antigen that's able to link up to both of these cells with their receptors, all right? So super antigen's not recognized by the receptor, but it is able to bind basically to both pieces of that non-specifically, right? Not in that active, or in that binding site. So as this happens, it's an uncontrolled activation of that T cell. And what happens in this case is these antigens link up, and what should be happening, forgetting about this not functioning, what should be happening is this guy comes in, interacts, and they make the exchange. Now the T cell's got it, and it does something about it, all right? It's not functioning like it should. And over here, the exchange isn't happening. They're locked together, all right? And that stimulates this T cell, our green guy down here, it stimulates that T cell to start releasing cytokines to say, bring in the troops, but it doesn't stop. It just keeps that mechanism turned on, dumping cytokines, all right? And as that happens, we see excessive, maybe uncontrolled inflammation. It's a process that is not something that, I mean, maybe there's a slight benefit, but overall, the level of inflammation, the issues that are resulting are more of a detriment to us than a benefit, right? And this is something that we can call a cytokine storm. As this is happening, you get a massive influx of these cytokines released, stimulating other cells to come in, and the enhancement of inflammation as the result of all of these other cells, and the possibility of more of this stuff occurring instead of the proper recognition compounds those ill effects. Crazy. So here we've got a B cell, we're only seeing a fragment. So B cell plasma membrane, and this is a receptor, B cell receptor that's on these membranes. These, as we mentioned, maybe it was last time, it could have been Monday, when we were talking about the different classes of immunoglobulins, the IgD, immunoglobulin D, is a B cell receptor. That's what this is, all right? This immunoglobulin is on the surface of these B cells, and it's got its specific binding sites that allow the B cell to recognize specific antigens in a similar fashion like the antibodies are doing their recognition, right? So these receptors bind specific antigens as this is happening. And that's what we're seeing here with, so these again would be those IgD antibodies or immunoglobulins, B cell receptors, right? So here we've got a bacterium and it's got some markers, fingerprints, if you will, on its surface, some polysaccharide antigens, and there's some epitopes, again, stimulating that immune response that this the receptor can recognize. As that happens, this is an independent, T cell independent activation. We don't need the T helper cell to tell this B cell what's going on. They've recognized it, all right? And this is, in this case, able to respond very rapidly as this recognition happens, all right? So it recognizes, activates, doesn't need those T cells, and now we start secreting IgM. So this pentameric immunoglobulin that has these binding sites that are exactly the same as the binding sites on this B cell that recognize this specific pathogen. It's producing antibodies that the B cell over here says, look what I found, make this exact thing, right? Now it secretes these into the plasma, into your circulation to float around and seek and destroy, right? They want to grab on to all of these pathogens and make these big agglutination complexes, stimulate other things to come in and deal with it, all right? So a second signal, as we say here, it's something that's not shown, the T lymphocyte receptors and the PAMPs, these little fragments on that bacterium are required to activate a B cell in the first place, right? But that's beside the point, all right? The T cell dependent, which is more of the classic type of humoral response, relies on these T helper cells, those CD4 cells, all right? So up above, we've got our B cell, the antigen linking up to that receptor, and in turn showing this fragment after we have dealt with it to a T cell, which in turn tells other B cells, not like this, other B cells to come in and see what's happening, and they differentiate into plasma cells and memory cells. It's not the same, but it's a similar idea and two mechanisms to try to mount as adequate of a response as you can. So this generates massive amounts of plasma cells, the antibody secreting factories. We see a lot of these immunoglobulins, in this case, probably IgG that's being secreted out, and then some memory cells laying in wait. And these memory cells could be what's snagging this stuff and coming right back up to the top for another response. So we showed at the start of this chapter, we showed an immune response that was a primary and secondary response, but we only showed one line as we moved through there. It was kind of the average of IgM and IgG together put onto that figure. You can monitor the specific immunoglobulins. You could look at the variation between these over the course of time. And if you separate them and plot them, you can see there are definitely some differences, right? Remember the IgM, we just showed that a second ago, is that pentameric form. The IgG is a monomer, a single one, all right? So IgM in blue is the first immunoglobulin that starts to get secreted upon recognition by those macrophages or other antigen-presenting cells. T cells get stimulated to tell B cells what's going on, and the first immunoglobulins that they start dumping out are IgM, that big pentameric molecule with the hopes that it's going to grab lots of things and start dealing with these invaders. All right. Shortly after our IgM, as it peaks and starts to wane, IgG is being the next class of immunoglobulin that's being pumped out, and it is a little more pronounced. It happens a little bit later, a little more pronounced, and it's also a little longer duration that it persists in our circulation. And again, this is the primary response. So our first exposure takes a little bit of time, maybe 2 weeks, to start seeing our first hint of a response. At about 3 weeks into it, we've got a pretty good IgG response, and then it starts to decline. We've got a little jig-jag in here because we've got a potential for a period of time that you hope goes before your next exposure. And as we mentioned previously, we could be talking vaccines or actual infection in these circumstances. That really is kind of beside the point. So after a period of time, we get another exposure to that antigen. We see IgM responding, and then the IgG. And in this instance, this response really should be maybe a little more pronounced right here. It's considerably more rapid. The idea, however, is the IgG. As we see it, that's the one that's the bigger player when we think about mounting an actual defensive mechanism. The amount of IgM is almost the same, right? It's kind of, if we were to put them together, about the same as the first primary response, but IgG is not. And it spikes considerably. A lot of this are the memory cells that we had generated previously. There are more of them to begin with, and you get a huge amount of IgG, and that is considerably elevated and persists much longer than the IgM response. This really the idea of lending to, with vaccinations and immunity over the course of time, what sets the stage for possible need for booster shots as the elevated level of IgG starts to decline. Depends on the type of vaccination. So I think we're close to the end of this chapter. There are a handful of different ideas as far as how we pick up some of our immunity in the first place. It could be naturally acquired or artificially acquired. And with each of those, it might be passive or it could be active, right? So 4 different ways, 4 different classifications as far as how we can pick up some of this immunity. The first one, the naturally acquired passive, we can see antibodies that are picked up to that infant, to that newborn. As we described, IgG has the opportunity to cross the placenta, imparting some of the mother's protective immunity, IgG molecules, to that child. And then also through breastfeeding, they can pick up immunoglobulins that way as well. And IgG and IgA can be acquired in those circumstances. Remember, the dimer form of IgA, the secretory form, would lend to some protective immunity that way. And it's more thinking benefits with like intestines as we're consuming that. It provides some protection that way. So natural but passive, artificial acquired passive immunity. This is like if you go donate plasma. When you donate plasma, they are taking your red cells and pumping those back into you. And the plasma, the fluid those cells are floating in, has all sorts of things. Immunoglobulins specifically is what they're looking for. All sorts of immunoglobulins that they can extract from you and separate and purify. With that said, they can use that as a passive mechanism to infuse you with some of that, and it confers some immunity, passive immunity. With that said, it is not like you've received a vaccination and produced these things on your own. You don't have all of the memory cells. You don't have the potential for these things to lay in wait and respond against another infection. This is something that is short-lived, all right? So the active natural is, come back to me, mouse. The active natural immunity is something that you get through dealing with an infection. So as you get that illness and recover, you've mounted an immune response like we've just described and produce those memory cells. That gives you this active immunity, natural immunity, and the ability to potentially respond later. And then the artificial active would be a vaccination where you are mounting that response, as opposed to this, where it's just the antibodies. Now you're producing those antibodies and keep those cells. It's a similar but different idea, all right? So that brings us to thinking a little bit about vaccination in the first place. Variolation for smallpox originated in the Far East, in China, and was spread. I think this is a Japanese image, but it is, yeah. It spread to Europe and Africa. Who was it? I think Lady Montagu was the one that had traveled, I believe, to Japan and witnessed some of these practices and brought these ideas back to Europe. It took a minute to kind of take hold, but it was something that held some promise, all right? Smallpox caused all sorts of issues, highly contagious. Lots of the wealthy would, they just leave and head up to the, outside, up to the hills and try to avoid all of these issues. Obviously, that's probably not so effective. Here we see this variolation. Basically, they would take an individual that is in the process of, dealing with smallpox. There's all of these pox-like lesions and scabs. They pick the scabs off, they grind those scabs up, and they blow that dried scab material right up the nostrils of an individual. You're blowing smallpox up their nose, all right? That is disgusting and disturbing. It conferred some immunity. It also gave people smallpox, all right? So what do you do, right? There are definitely issues with that, all right? Jumping forward a bit, Edward Jenner comes up with a different idea, and vaccination. Vaccinia is cowpox, and cowpox and smallpox are cousins, if you will, all right? He recognized that the milkmaids, the ladies out there milking cows, didn't get smallpox. and he started to wonder what's going on. He recognized that those ladies, the milkmaids, got a different pox-like infection, the cowpox. They picked that up from being in close contact with the cattle, and the infection with cowpox, which was definitely more mild, obviously conferred some protection, he figured, against smallpox, but he wasn't sure, all right? So he basically got a child. I have heard multiple stories. My first understanding was it was his own son that he used and he inoculated. So took some of this and injected into the child, and then later exposed the child to smallpox, and they didn't develop it. And there we've got our vaccination. I have later heard that it wasn't his kid. It was just an orphan. It was easier, you know, it's like a lab animal. Just go down to the orphanage. You can buy yourself an orphan and then you can do some science experiments on it. That's terrifying, right? And here we are today, right? So ultimately, fortunately, that child didn't develop smallpox as he was exposed to that. And there we go. I will leave you with that. Farewell. Have a great weekend.